Hello everyone, I'm David and I study the effects of silver nanoparticles on spore germination, gametophyte size, sexual differentiation, and sporophyte root growth in the fern Ceratopteris richardii. A brief overview of what silver nanoparticles are. They are widely used in commercial products in agriculture due to their antimicrobial and antifungal components. However, the release of nanoparticles into the environment can pose a potential hazard to biological organisms. Studies have shown that the effects of silver nanoparticles on flowering plants are complex and can vary depending on factors such as nanoparticle concentration, plant species, exposure duration, and environmental conditions. Few investigations have looked at the influence of silver nanoparticles on non-flowering plants such as mosses and ferns. Our study specifically investigates the impact of silver nanoparticles on the model fern Ceratopteris richardii, which is a homosperous fern, which produces one spore that will then differentiate into a distinct hermaphrodite or male gametophyte by hormonal sex determination. To begin our study, we sown the spores. We used an agar in a nutrient solution supplemented with the silver nanoparticles, ranging from 0 to 100 parts per million, 0 being our control. The plants were maintained at a stable temperature and humidity, and we used the compound light microscope and SEM to take our images, which was most of our data. Data was collected on spore germination rates, ratio of hermaphrodites to male gametophytes, relative gametophyte size, and sporophyte roots following fertilization. Spore germination rates were analyzed using an ANOVA, a one-way ANOVA, and a t-test. Moving into our results, Figure 1 shows spore germination at 5 days from sowing. We can see that our concentrations, our treatments from 20 to 100, are not significantly different from our control. Figure 2 shows spore germination again, but this time at 14 days from sowing. Like in Figure 1, we can see that our treatments are not significantly different from the control, with the exception of 100 parts per million being significantly different. Moving on to Figure 3, we have our sex ratios. We found that hermaphrodites compared with males are generally less across concentrations, almost half. In Figure 4, we have gametophytes at 26 days from sowing. Photo A shows a healthy, mature hermaphrodite that has green tissue and it's large. In photo B, we have a hermaphrodite that is delayed in growth, although it is still green, and this is at 20 parts per million. In photo C, we have two mature hermaphrodites, with the larger being our control and the smaller being our 40 parts per million. From viewing this, we can see that the treatment is visibly smaller than our control, and this remains true across all concentrations. In photo D and G, we have two mature healthy gametophytes, with D being our mature hermaphrodite and G being the male gametophyte. There are very few that were found like this. That's why in Figure 3, our numbers are low, because most of them look like photos E, F, H, and I, which are immature, lacking chlorophyll, so they became chlorotic and transparent, and then eventually died. Figure 5 just shows normal gametophyte development in C. richardii. This illustration is from the literature. Moving to Figure 6, we also observed abnormalities in the gametophytes. Photo A shows their white arrow is pointing to a definitive meristematic notch, which is characteristic of the female hermaphrodite. But we also have the black arrow, which is pointing to an abnormal cluster of male antheridia, the male sex organs, which is in an unlikely spot. Moving to photo B, we have another cluster of healthy male antheridia, with the rest of the tissue becoming chlorotic and ultimately dying as well. In figure C, we have an undifferentiated chlorotic and necrotic gametophyte. We weren't sure if it was going to be a hermaphrodite or a male because it wasn't distinctive of either one. Moving to figure 7, after fertilization, in figure 7 and figure 8, we looked at sporophyte root tips and sporophyte length. Photo A in figure 7 shows 0 parts per million healthy sporophyte root tip. As compared to photo B in figure 7, we have 40 parts per million that has a necrotic sporophyte root tip that has become brown and is dying. Moving to figure 8, we have our SEM photos of our sporophyte roots and sporophyte length. In figure A, we can see this is a healthy sporophyte root tip. It is starting to slough off that outer cells, and then that will allow it to begin growing and continue that process. In photo B, we also have the sporophyte root tip, but as you can see, it is severely damaged. In photo C, we have our sporophyte length, and it's the same photo as photo A, and it is in the same concentration. But it is important to note that photos C and D in figure 8 are taken at 31 days. These are very healthy in photo C, and they are developing naturally as they should. In photo D, we have a very, very small sporophyte root. It is hard to see, but it is delayed in its growth, and it is very abnormal. In our discussion in conclusion, we found that all concentrations of silver nanoparticles from 20 to 100 ppm had varying negative effects on the growth and development of C. richardii. Our study is important because it supports recent findings that silver nanoparticles released into the environment can potentially have negative effects on the ecosystem. Spore germination across treatments was on par with those of the control. However, spore germination in Ceratopteris, a similar fern in the Ceratopteris family, decreased as the concentration of AgNP silver nanoparticles increased. However, other studies have shown that silver nanoparticles can have a beneficial effect on seed germination at low concentrations due to their antimicrobial and antifungal properties. Silver nanoparticles significantly alter gender differentiation by reducing the number of hermaphrodite gametophytes across treatments. Another study obtained similar results in, again, Ceratopteris, a similar fern, that reduced numbers of hermaphrodites within populations would reduce the number of offspring. We observed our gametophytes, and they were delayed in growth compared to the controls across all concentrations, as shown in figure 4. Hermaphrodites were smaller than those of the control, and a decrease in plant biomass is very common under stressful, contaminated environments, once again as shown in figure 4. By day 26, gametophytes found in the higher concentrations, that being 60 parts per million to 100 parts per million, suffered chlorosis, which means they lost their chlorophyll and ultimately died, became necrotic before reaching maturity. Roots had damaged root tips and they were stunted in their growth compared to that of the control. It's important to note that soil pollutants like metal nanoparticles and microplastics inhibit root growth in a wide range of plants. While some studies show potential benefits of silver nanoparticles at lower concentrations, caution is warranted due to the potential toxic effects at higher concentrations. Therefore, further research is needed to fully understand and grasp the mechanisms and long-term consequences of silver nanoparticle exposure on plant growth and the ecosystem at large.